So today we're going to look how we can culture microorganism in the laboratory. So we need to culture microorganism in the lab for various purposes. Now the question is, can we culture all the microorganisms? Well, answer is no. We can't culture all the microorganisms. Why is that? Because certain microorganisms, they have a very special habitat. Even it's a microhabitat, it provides special conditions. So if we can't provide this condition, then we cannot grow microorganism in the lab. So we need to establish right specification and method to grow some of the microorganism. But we can grow certain microorganism which we need to study and certain other purposes which are already established this method. Now here, what is very important to understand, what is the growth medium of microorganism? So when you talk about the growth medium, so we have to first understand what is a growth medium for bacteria. That's very important, bacteria. And maybe if you want to grow fungi, what is the growth medium for fungi? And also, if we still want to know a little bit about, can we grow virus in the lab? Yes. How we can grow virus in the lab? What is the growth medium? Okay, first we will look what is the growth medium for bacteria. The growth medium for bacteria, the most available growth medium is called nutrients agar. So what is nutrient agar? So nutrient provide the required nutrition for the bacteria, and the agar provide the solid surface. So nutrients, what are the nutrients? It has beef extract, yeast extract, and peptone, and sodium chloride. All right, now if we get the right amount and then add water and then dissolve it, and after that we have to use a suitable sterilization method. So for example, autoclaving, 121 Celsius degree, close to 100 kilopascal high pressure, 30 minutes, enough to sterilize it. Right, now let's quickly look how we can really grow this how we can prepare a medium to grow the bacteria. So if you look at this video, so this is the powder form which is available commercially to grow the bacteria. This is slightly different one. It's called brain heart infusion agar. So first step, we have to weigh a certain amount of the powder into a container. And after that, we can add water, right amount to this powder. And then what we can do, we can dissolve it. So let's see how we can do it. Now look at here. In this video, you can see the powder is adding to the weighing board, and roughly around 5 grams, taking into this conical flask, and then adding water, and we use this magnetic stir to dissolve the agar inside the water. Right, after that, we can use an aluminum foil to cover it, and we can use this indicator tape to check whether sterilization is properly happened, because after sterilization we can see a black stripe on this tape. So this is the autoclave. So this is used to sterilize it. So you can add the water to make the vapor, and you can use this the container to place this container, and then you can close it properly to maintain the pressure. After that, you can indicate the suitable temperature, 121, 17 minutes, and machine automatically adjusts the other parameters, and then after that you can take this carefully, and you can see this black stripe here. You can see this black stripe on it here that indicate it's properly sterilized. Right now, coming back to the other mediums, we can also use another medium which is called potato dextrose agar medium. So it has potato, and then in potato infusions, and then a dextrose, kind of a glucose, and then this is also another important medium. So it's a potato dextrose medium. So potato dextrose agar. Fine. So we have learned here how we can make a medium for bacterial growth. Before I wind up, we have to grow bacteria sometimes not only a solid surface, we have to grow bacteria on a floating conditions. So for that we use nutrient broth. It's nothing much. We don't add agar to solidify. Then we can grow bacteria on this in this nutrient broth. Okay, so next we're going to look how we can grow fungi in our labs. So I just delete this one. To grow fungus, we use mediums, for example, malt extract and brain heart infusion medium. So you have seen in the video, brain heart infusion medium. And there's a special medium we can rely on mostly. It is a Sabarode dextrose medium. So Sabarode dextrose medium is suitable medium to grow fungus in the lab. So this Sabarode dextrose medium contain dextrose is glucose, D-glucose, peptone, agar, distilled water, and that's it. So dextrose, peptone, agar, and distilled water. Dextrose, peptone, agar, and distilled water. Here you see we don't want to add the sodium chloride because dextrose can manage this osmotic balance. So no need to add sodium chloride. That means we don't add sodium chloride always, like you have seen in nutrient agar medium, we add sodium chloride. All right, so last, just to know a little bit about that, virus. You know, virus cannot grow in outside because they don't have a complete machinery to do protein synthesis and other functions. So they have to hijack a host system to grow. As a result of that, we need a live cell to grow virus. Either we find live cells, or maybe we can get animals, infuse animal injection into the animals, providing the live host to grow virus. Okay, so That's it. To know about the growth medium, let's have a look this question 2019 paper about the growth medium. Which of the following statement is correct regarding the culture media? Media is a plural, medium is singular, used to grow microbes. So microbes essentially, all the microorganism, bacteria, virus, prototis, and fungi in the lab. So we understood the question. What is correct sentences to grow microorganism in the lab? So number one, Agar in culture media provides the suitable pH range for the growth of microorganism. Now I told you, Agar provides the solid surface to grow microorganisms. And you know, pH we can adjust by adding buffers. So buffer solutions does it, but not the agar. So this is wrong. Glucose is generally used to prepare culture media to grow fungi. Well, I like these terms like a generally, because when the questions are using very extreme terms, you have to be a little bit careful, but they are not that extreme. You have to be a little bit think about these questions. Okay, we have seen in our video what is the growth medium used for fungi? Saccharode dextrose medium. So saccharode dextrose medium, essentially dextrose is a glucose. Well, then we can a little bit of agree with this answer. Generally, we can add glucose to grow fungi. Third one, culture media for bacteria prepared using potatoes. Culture media for bacteria prepared using potatoes. Okay, this sentence is one hand yes, one hand we can say no. Prepared using potatoes. So we have seen potatoes, dextrose, agar. That means we use potato extracts to grow the bacteria. So then you can say, well, park this choice and go ahead. Any microorganism can be cultured in culture medium. Well, I told you from the beginning, I asked you a question, can we grow any microorganism? I said no. So then we can remove this one. Sodium chloride is usually added to all culture media. Okay, they use the word usually. That means, well, it's a generally we can add. But we have seen all culture media. Look at my video. Saccharode dextrose area has no sodium chloride. Then with all culture media, we don't want to add. So we can also remove this one. So now choice 2 and 3 left. Then this is a little bit of too harsh. We use potatoes, but you know, we can't boil the potatoes and then get this extract, and then we use like this kind of you know, homemade kind of impression you get to use the culture media. I think we remove this one, and also we prefer the second choice. Generally, we have seen to grow fungi, dextrose is added. So we can take choice 2 is the most appropriate for these questions. Okay, I wind up this video. So we will meet again for next lesson for GCE A-level biology in the next time. So hope you enjoyed the video and see you again next time.